Hello students, today I am going to discuss one of the important topic and it is biomedical waste management. Firstly, we will see the introduction of biomedical waste management, and then we see the definition of biomedical waste management, and after that we have a measures for waste minimization and various names for biomedical waste, and after that we have a classification of biomedical waste management and what are the steps for the biomedical waste management, and in the last we have a cross infection, what are the types and the causes of cross infection and its treatment and its precautions. So now let's start with the introduction part. In this contemporary world, the disease rate are considerably high. As we know that the disease rate are very high, and if the disease rate are high, the disease management rate are also increased. So I think the biomedical waste is an alarming issue nowadays. So biomedical waste is a threat for man, community and for the environment, as you can see the diagram of hospital waste management. Then we have a definition of biomedical waste. What is biomedical waste? Biomedical waste is the waste which is generated during the medical research, testing, diagnosis and immunization and the treatment of human beings or animals. In simple words, we can say that the waste which is generated in the hospital during treatment is called a biomedical waste, and these biomedical waste are put in different containers according to their categories. As you can see the diagram here, red dustbin, yellow dustbin, blue dustbin and a white dustbin. So then we move to the second slide. What are the measures for waste minimization? Firstly, we have to use the reusable items which made of glass and metals, and second point is select the non-polyvinyl chloride plastic items, and the third step is we have to adopt the proper policies and proper management of waste generated. So we have to segregate the waste according to their priority, and last measure is we have to recycle the policy for recycling waste. Then after that we have a different names for biomedical waste management, and it include clinical waste, medical waste, biohazardous waste and infectious waste management, regulated waste and a healthcare waste. And after that we have a classification of biomedical waste. Biomedical waste is divided into the three categories: non-infectious, infectious and pathological. Firstly, the non-infectious waste is 70 to 75% to 80%. Infectious waste include 15%, and hazardous category include 5 to 10%. According to the WHO has classified the hazardous waste in the different categories like infectious. As you can see, the infectious waste include lab cultures, isolated wards, tissues, swabs, materials and equipments of the infected patient. As you can see in the diagram that there are some swabs are also seen in the diagram. Then after that we have a pathological waste. Pathological waste include human tissues, body parts and body fluids which are collected in this containers. After that we have a sharp waste, and the sharp waste include needles, infusion sets and knives, blades and broken glasses, as you can see in the diagram. After that we have a pharmaceuticals related to drugs. If we have a expired pharmaceuticals or a banned pharmaceutical drugs, so this is also a category. After that, we have a genotoxic waste. Genotoxic waste includes cytotoxic drugs and which is used in a cancer therapy, and genotoxic chemicals. After that, we have a sixth category, chemical waste. Chemical waste include lab reagent, expired disinfectant, and expired solvents, as you can see in the diagram. So after that, we have a waste with high content of heavy metals. It include batteries, broken thermometers, and after that we have a pressurized containers, gas cylinders, and aerosol cans. And the last category is radioactive waste. It include lab research liquids and contaminated glasswares, packages, and absorbent papers. So after that, we have a steps for waste management. Firstly, we have focus on 4 R's mainly: reduce, reuse, recycle, and recover. After that, we have focus on the 4 main steps of waste management. In the first step, we segregate the waste in a color coded bags at the point of generation. We segregate the waste in the first step. We segregate the waste in the different color coded bags at the point of generation, and in the second point, we collect and store the waste in a CSSD, central storage area, in the hospital. After that, we have a third step, and third step include transportation. In this step, transport the waste into the authorized facilities. And in the fourth step, we have a treatment and disposal within the 24 hours of waste generation. Then there are various steps in the manage the segregation of waste. Firstly, we can see that we use the different dustbins for different waste. Firstly, the yellow dustbin is used for anatomical waste, chemical waste, and a solid waste. Mainly, yellow dustbin is mainly used for dressings. Infectious material is put in that dustbins, like human body tissues, solid waste, discarded linens, and laboratorical waste. After that, we have a red dustbin. In the red dustbin, we mostly used contaminated plastic waste. We mostly put the contaminated plastic waste, and it include tubings, plastic bottles, intravenous tubes, catheter, cannulas, all the material which made from the plastic. Contaminated plastic are put in the red dustbin. And this question is very important as exam point of view. So now let's move to the next slide. And next is third one is a blue dustbin. Blue dustbin is in the blue dustbin we put the glassware such as broken or discarded vials, bottles, and slides. And the last one is a white dustbin. White dustbin are used for a sharp needles and blades, and it is a puncture proof container in which we discard the needles and blades. Then we have a transportation and storage step. We have the waste is temporarily stored at the central storage area in the hospital. This one, central storage area. The waste is mainly stored in the central storage area in the hospital, and after that the central storage area in the hospital, then it's sent to the final disposable once or twice a day. It totally depends on the quantity of the waste. And during transportation the following points should be taken care of. And firstly, the bags and containers are properly sealed or labeled. And second one is, bag should not be filled completely. if the bag is filled completely, then it may a chance of broken or some spillage on the way. So it's better to use to be half filled. Then the transportation, during the transportation, the site of the final disposal of these waste are done via motor vehicles, trucks, tractors and trolleys. And after that, as you can see in the diagram, biohazardous symbol, this is a symbol. Vehicles used for transportation of biomedical waste must have a biohazard symbol, and these vehicles should not be used for any other purpose. And as you can see in this cytotoxic hazard symbol, which is included, which is mostly used in a cancer patients and cancer therapies. And after that, the label should be clear and prominently visible and non-washable. After that, we have a disposal of biomedical waste management. There are different methods used for it, and one of them is deep burial. And in this trench, the dug is 2 meter deep, and it should be half filled with the waste, and it covered with the lime and 50 centimeter of the surface, and then we put some soil over it, and we must ensure that animal don't have any access to that particular site. And second method is a autoclave. This is also as you can see in the diagram, it looks like a pressure cooker. And autoclave is a machine which is used to kill the microorganism, harmful microorganism, bacteria, virus and fungi and spores on the items. It kills the harmful microorganism, and it is these items are heated for a particular point of time, particular amount of time. Then after that, we have third one, incineration. Incineration is also used for burning the biomedical waste. Initially, the waste is put in the primary chamber, as you can see in the diagram, and then the temperature reaches 850 degrees Celsius, and then the gas are released from the secondary chamber, and the incineration process is completed. And then we studied the personal protective equipment. Always wear a personal protective equipment while handling the biomedical waste. And as you can see in the diagram, we should always wear mask, face shield, goggles, cap, gloves, boots and gowns. These all are the biomedical personal protective equipments. And after that, we have taking a immunization against hepatitis B and tetanus are very important. And what is cross infection? Cross infection is the transfer of harmful microorganisms such as bacteria and virus which spread from one person to another with the help of equipment or any other object or within the body, and this infection may cause serious complications. For example, in a medical ward, the medical practitioner checks the 10 patients, and he or she doesn't use the hand rub or don't do any kind of hand washing. At that time, she or he may transfer the cross infection to the other patients. Then what are the types of cross infection? There are many different types of cross infection occur. Firstly, we have a urinary tract infection. In this, urinary tract infection is caused mainly by the catheters. Catheters are inserted mainly in a long-term patients, surgery patients. After that, we have a surgical wound infection. Surgical wound infection may cause redness, swelling and pus, and sometimes patient may have a fever, high fever, and infection related to the presence of peripheral or central venous access line. These lines are mainly used in the chronic patients or ICU patients. So in these lines the patient are more prone to the cross infection. Then next we have a causes of cross infection. What are the causes of cross infection? There are bacteria, fungi, virus and parasites are the main cause of cross infection. But there are also some other reasons like unsterile medical equipments. If in the hospital the medical staff use the unsterile medical equipments in the patient used for the patient surgeries, then it may cause cross infection from one patient to another. And by coughing and sneezing we transmit the cross infection, and by human contact like touching, hand shaking, etc. And touching contaminated object. If the object is contaminated and we touch that contaminated object, then we also transmit the cross infection. And the dirty bedding is also one of the cause for it. And last one is prolonged use of catheter, tubes and intravenous lines are the causes of cross infection. And what are the preventions or precautions to be taken while handling the patient? Firstly, isolate the patient, restrict the visitors, maintain the space between the two beds, and medical staff must maintain the aseptic techniques, and proper mask, gown, gloves, goggles are also applied by the medical practitioners, and perform a hand hygiene before and after every patient contact. Use and dispose of shafts safely, and perform the routine environmental cleaning. Use proper aseptic techniques, and handle and dispose of the waste used in linen safely. These are the precautions we should take in a hospital. Then after that we have how to prevent the cross infection. In the cross infection we see that we prevent it by use the aseptic technique. If we use the aseptic technique in the hospital, then we prevent the cross infection. If we use the sterile equipments, so it may prevent the cross infections. Or in the hospital during the procedures we use proper aseptic techniques, so we don't transfer the infection from one person to another. And after that we have a hospital should be fully ventilated. As you can see in the diagram, the hospitals should be fully ventilated, and proper space between the beds are also available in the hospitals, and proper aseptic techniques are followed by the medical staffs. And in the hospital, masks should be in the infectious areas, the mask should be compulsory for our medical practitioners and for our patients as well as. And last step is maintenance of general cleanliness of the hospital. In the hospital, if the hospital is not cleaned properly, then it may cause infection to the patient as well as for the medical. It is also harmful for the medical staff as well as. So now that's it. So now let's summarize the topic. Firstly, I discussed the introduction part of the biomedical waste management, and after that which measures for waste minimizations are used, and what are the various names of biomedical waste management and its classification of biomedical waste management, and what are the steps of biomedical waste management. And the last question is, what is cross infection, what are the causes of it, and how it treated, and what are the preventions and precautions should be taken by the staff. Thank you.
स्टूडेंट्स, आज हम पढ़ेंगे बायोमेडिकल वेस्ट मैनेजमेंट के बारे में कि बायोमेडिकल वेस्ट होता क्या है, उसे कैसे मैनेज करते हैं एंड क्या डिफरेंट डिफरेंट कोड्स हैं जिनके अकॉर्डिंग हम वेस्ट जो हमारा बायोमेडिकल वेस्ट है उसको हम सेग्रीगेट करते हैं। सो लेट्स स्टार्ट टुडे क्लास। द टॉपिक इज बायोमेडिकल वेस्ट मैनेजमेंट। ओके, फर्स्ट ऑफ ऑल व्हाट इज बायोमेडिकल वेस्ट? बायोमेडिकल वेस्ट होता क्या है? एनी वेस्ट, वो कोई भी वेस्ट जो कि जनरेट होता है, प्रोड्यूस होता है, कब? आपका डायग्नोसिस के केसेस में। ओके, डायग्नोसिस, ट्रीटमेंट और इम्यूनाइजेशन ऑफ ह्यूमन बीइंग, करेक्ट, और एनिमल्स। सो ऐसा कोई भी वेस्ट जो प्रोड्यूस हो रहा है, जनरेट हो रहा है, कब? जब आप किसी पेशेंट का इलाज कर रहे हो। दैट मीन्स कि आपके डायग्नोस्टिक टूल्स हो गए, आप एक्सपायर्ड मेडिसिन हो गई, या जैसे आपने डायग्नोस में आप ये कह सकते हो कि ठीक है, जैसे कुछ आपके सैंपल्स होते हैं, लाइक ब्लड सैंपल टेस्ट किया, तो अब वो जो सैंपल टेस्ट करने के बाद रिमेनिंग सैंपल है, दैट इज योर वेस्ट। ओके, एंड लेट सपोज नेक्स्ट एग्जांपल यूरिन सैंपल है। यूरिन सैंपल को आपने डायग्नोस किया। ओके, सो आफ्टर द एग्जामिनेशन ऑफ दैट सैंपल, अब वो सैंपल यूज हो चुका है तो अब वो डिस्कार्ड करना पड़ेगा, तो वो आपका बायोमेडिकल वेस्ट है। वो एक डायग्नोस्टिक वैल्यू से जनरेट हुआ है। एंड सेकेंड इज ट्रीटमेंट। ट्रीटमेंट से जैसे आपका, जब हम ट्रीटमेंट देते हैं, कीमोथेरेपी देते हैं कैंसर पेशेंट्स को, या फिर नॉर्मली हम देखें जैसे मेडिसिन देते हैं, ठीक है, वैक्सीन देते हैं, इम्यूनाइजेशंस में आपके सिरिंजेस हो गए, इंजेक्शंस हो गए, आपके जो वेस्ट होते हैं वो भी वेस्ट प्रोडक्ट है, जो कि एक ह्यूमन बीइंग या एनिमल को ट्रीट करते वक्त, इम्यूनाइजेशन देते वक्त वो वेस्ट प्रोड्यूस हुआ है। तो कोई भी ऐसा वेस्ट जो कि लिविंग थिंग्स, बायो, एक्चुअली द वर्ड इज बायो, बायो मीन्स लिविंग, ओके, एंड मेडिकल। सो मेडिकल मीन्स कि मेडिकल हेल्थ सेक्टर से रिलेटेड जो वेस्ट है, कोई भी वेस्ट जो लिविंग थिंग से रिलेटेड होगा, फिर चाहे वो उसके डायग्नोसिस से रिलेटेड होगा, ट्रीटमेंट से, या उसकी वैक्सीनेशन से, किसी भी ह्यूमन बीइंग, एनिमल, लिविंग थिंग में तो सब कुछ आता है, या फिर उसकी कुछ रिसर्च एक्टिविटीज से जो भी वेस्ट प्रोड्यूस होगा उसे हम बायोमेडिकल वेस्ट कहते हैं। रिसर्च एक्टिविटीज लाइक जैसे रिसर्च सेंटर होते हैं कि वहाँ पे जो रैबिट्स होते हैं, हमारे सकलिंग माउस होते हैं, ओके, जो उन पे रिसर्च होती है, जो हमारे रिसर्च एनिमल्स होते हैं, या फिर वहाँ पे रिसर्च के लिए जो मेडिसिन, जो रिएजेंट्स, जो भी चीजें यूज करी जा रही है, और उस रिसर्च के टाइम पे जो वेस्ट प्रोड्यूस हो रहा है वो हमारा बायोमेडिकल वेस्ट है। सो क्लियर विद द डेफिनेशन ऑफ बायोमेडिकल वेस्ट कि बायोमेडिकल वेस्ट हम बोलते किसे हैं। सो इन सिंपल वर्ड्स, बायोमेडिकल वेस्ट are the waste that produce from living beings. Okay, living beings are your, from living beings, so living beings are your animal, maybe human, maybe, okay, anything that is related to medical, that is your biomedical waste. Next, categories of waste. There are different categories of waste. अगर हम कैटेगरीज में डिवाइड नहीं करेंगे तो हम उसको ट्रीटमेंट नहीं कर पाएंगे, क्योंकि बायोमेडिकल वेस्ट जब हम इकट्ठा हो जाता है एक जगह पे, उसके बाद हम उसको ट्रीटमेंट करते हैं प्रॉपर। जैसे हमारे जो हमने पढ़ा होगा, हमेशा से सुना होगा आप लोगों ने भी कि रीयूज करो चीजों को, रिसाइकल करो। ओके, सो वो कैसे होती है रिसाइकल? जब जो वेस्ट चीजें होती है उनको पहले ट्रीटमेंट करा जाता है, प्रॉपरली उनको डिसइंफेक्ट किया जाता है, स्टेरलाइज किया जाता है, उसके बाद वो चीजें रीयूज होती हैं। सो कैटेगरीज है कि हमें कौन सा वेस्ट कहाँ पे डिस्कार्ड करना है, और फिर उस पर्टिकुलर वेस्ट को हम कैसे ट्रीट करते हैं, ठीक है। जैसे कि अब आप, ऐसा नहीं है कि कोई ब्लड से इन्फेक्टेड चीजें है वो भी आप एक जगह डाल रहे हो, शार्प चीजें भी एक जगह डाल रहे हो, ओके, एंड जो आपकी इन्फेक्टेड चीजें हैं वो भी एक जगह डाल रहे हो, डिसइंफेक्टेड चीजें भी एक जगह डाल रहे हो, जनरल वेस्ट एक जगह डाल रहे हो, मेडिकल वेस्ट एक जगह डाल रहे हो। नो, दैट इज नॉट अ करेक्ट मेथड फॉर डिस्कार्डिंग द वेस्ट। हमें एक कैटेगरी बनाई गई है, WHO से गाइडलाइंस है कि हमें कैसे, किस तरह से, कौन से कंटेनर्स में, कौन से कलर कोड्स में, उन कलर कोड्स का क्या मीनिंग है, आपको किस तरह से वेस्ट को डिस्कार्ड करना है, ये सारी चीजें सीखेंगे हम। हमारे पास जनरली येलो कलर कंटेनर्स होते हैं, कैटेगरीज डिवाइड है। सो येलो कलर होता है व्हाइट कलर होता है दैट इज ट्रांसपेरेंट, यू कैन सी ब्लू कलर होता है, एंड रेड कलर होता है, ब्लैक कलर होता है, एंड ग्रीन कलर्स होता है जो हम अपना ऑर्गेनिक चीजों के लिए यूज करते हैं। ओके, जैसे सो फर्स्ट ऑफ ऑल वी टॉक अबाउट येलो। येलो कैटेगरीज के जो बैग्स होते हैं, जो कंटेनर्स होते हैं, वो नॉन क्लोरीनेटेड प्लास्टिक्स यूज होती हैं उसमें, उन बैग्स में वो नॉन क्लोरीनेटेड होती हैं, दैट मीन्स उनमें क्लोरीन प्रेजेंट नहीं होता है। ओके, सेपरेट कलेक्शन सिस्टम होता है ट्रीटमेंट के लिए। इन जो येलो बैग्स होते हैं इनके लिए जो इनका ट्रीटमेंट सिस्टम है वो सेपरेट होता है। लाइक कुछ हम वेस्ट ऐसे हैं जिनको हम सीधा ऐश की फॉर्म में कन्वर्ट कर देते हैं। ओके, इंसिनरेशन से। कुछ हैं जिनको हम पहले स्टरलाइज करते हैं ऑटोक्लेव हॉट एयर ओवन की हेल्प से। इस तरह से डिफरेंट डिफरेंट कैटेगरीज को ट्रीट करने के लिए डिफरेंट डिफरेंट मेथड्स हैं। इसलिए हर एक कैटेगरी के लिए एक स्पेसिफिक है कि येलो कैटेगरी में आपको कौन सी चीजें डिस्कार्ड करनी है, रेड में कौन सी चीजें डिस्कार्ड करनी है, उसी के अकॉर्डिंग उसका कलेक्शन होता है आगे, एंड उसका फिर ट्रीटमेंट होता है। यस, वन मोर थिंग, जो बायोमेडिकल वेस्ट होता है उसको कलेक्ट करने के लिए हमारे पास दो सर्विसेज होती है। फर्स्ट ऑफ ऑल CBMWTF, दैट इज योर कॉमन बायोमेडिकल वेस्ट ट्रीटमेंट फैसिलिटी। यहाँ पे आपका जितना भी कॉमन बायोमेडिकल वेस्ट है वो ट्रीटमेंट उसको दिया जाता है। एंड सेकेंड वन इज HCF, दैट इज योर हेल्थ केयर फैसिलिटी। जो वेस्ट हेल्थ केयर सेक्टर से जनरेट होता है वो HCF में आपका ट्रीट होता है। ओके, सो कमिंग बैक टू अवर कैटेगरीज। सो फर्स्ट ऑफ ऑल येलो कैटेगरी। येलो कैटेगरीज के बैग्स हमने देखा कि वो नॉन क्लोरीनेटेड होते हैं, उनमें क्लोरीन प्रेजेंट नहीं होता। ओके, एंड जो येलो कैटेगरी होती है उसमें हम कौन कौन से वेस्ट डिस्कार्ड करते हैं? ह्यूमन एनाटॉमिकल वेस्ट, एनिमल एनाटॉमिकल वेस्ट, सॉइल्ड वेस्ट, expired or discarded medicines, chemical waste, micro, other clinical lab waste, chemical liquid waste. Okay, so let's start with human anatomical waste. Human anatomical waste वो होता है जो भी ह्यूमन से जनरेट हो रहा है। लाइक जैसे ब्लड बैंक का अब एग्जांपल लेते हैं, जब हम वहाँ पे ब्लड बैग्स लेते हैं, डोनेशन लेते हैं, तो वहाँ पे जो सेगमेंट्स होते हैं, वहाँ पे जो ब्लड हमें डिस्कार्ड करना है दैट इज द ह्यूमन एनाटॉमिकल वेस्ट। ओके, जो कॉटन्स है उस टाइम पे, ऑर्गन्स होते हैं, जो ऑर्गन्स डिस्कार्ड करते हैं हम हिस्टोपैथोलॉजी में। तो इस तरह से ह्यूमन वेस्ट इसमें जाता है, एनिमल वेस्ट जाता है। एनिमल वेस्ट कहाँ से आपका प्रोड्यूस होगा? रिसर्च सेंटर से बेसिकली, जहाँ पे रिसर्च होती है वहाँ पे एनिमल्स को यूज किया जाता है रिसर्च पर्पसेस के लिए। तो उस केसेस में आपका एनिमल एनाटॉमिकल वेस्ट होता है। एक्सपायर्ड मेडिसिन जो एक्सपायर्ड हो चुकी है या हमें डिस्कार्ड करनी है। ओके, केमिकल वेस्ट आ गए आपके रिएजेंट्स एंड ऑल। माइक्रो लैब होती है हमारी पैथोलॉजी, जो डिफरेंट डिफरेंट लैब्स होती है उनमें हम इन्हें यूज करते हैं। एंड केमिकल लिक्विड वेस्ट को डिस्कार्ड करने के लिए यूज करते हैं। ओके, नाउ नेक्स्ट इज योर रेड बैग्स। रेड बैग्स ऑल्सो आर नॉन क्लोरीनेटेड, इनमें भी क्लोरीन प्रेजेंट नहीं होता। रेड बैग्स में हम वो कंटामिनेटेड वेस्ट डालते हैं जो रिसाइक्लेबल होते हैं। ओके, सो द पॉइंट विच यू हैव टू रिमेंबर इज दैट waste we discard in the red bags are recyclable. that means कि हम उन वेस्ट को दोबारा से रिसाइकल कर सकते हैं, रीयूज कर सकते हैं। कौन कौन से हम वेस्ट डालते हैं? ट्यूबिंग, जो ट्यूब्स होती हैं हमारी, बॉटल्स, आईवी ट्यूब्स एंड आईवी सेट्स, कैथेटर्स, यूरिन्स बैग, जो यूरिनरी बैग्स होते हैं जो यूज करते हैं पेशेंट्स के लिए, यूरिन कलेक्शन के लिए। एक्चुअली किन पेशेंट्स में यूज करते हैं? जो पेशेंट्स मूवमेंट की कंडीशन में नहीं होते, जो प्रॉपरली बेड पे ही पेशेंट होते हैं, उन केसेस में हम यूरिन बैग यूज करते हैं, तो वो भी रिसाइक्लेबल होते हैं। एंड सिरिंजेस, यस, हम सिरिंजेस को भी डिस्कार्ड करेंगे बट विदाउट निडल। निडल इज अ शार्प वेस्ट, तो शार्प वेस्ट को हम रेड बैग में डिस्कार्ड नहीं करेंगे। दैट इज द पॉइंट which I am highlighting. Okay, so syringes करेंगे but without needle, because syringes जो है वो reusable है, वो उनको properly treatment के बाद reuse किया जाता है। treatment अभी हम देखेंगे कैसे किस बैग का करते हैं। and gloves भी हम discard करते हैं रेड बैग में, करेक्ट। सो दिस इज अबाउट रेड बैग। रेड बैग में हम क्या क्या डिस्कार्ड करते हैं? am I clear with that? कंटामिनेटेड वेस्ट करते हैं वो कंटामिनेटेड वेस्ट जिसे हम रिसाइकल्स कर सकते हैं। ओके, सो एग्जांपल्स आपने देख लिए, ट्यूबिंग, बॉटल्स, आईवी ट्यूब्स, आईवी सेट, कैथेटर्स, यूरिन बैग, सिरिंजेस एंड ग्लव्स। ओके, वन मोर थिंग, लेट्स गो टू प्रीवियस स्लाइड। ह्यूमन एनाटॉमिकल वेस्ट में हम बेसिकली इसमें येलो बैग में हम वो चीजें डिस्कार्ड करते हैं जो कि कंटामिनेटेड होती है ब्लड से। ओके, लाइक कॉटन, कॉटन अगर कंटामिनेटेड है ब्लड से तो वो येलो बैग में डिस्कार्ड होगी, राइट इसी तरह से एनिमल एनाटॉमिकल वेस्ट, मैंने आपको बताया था कि जो एनिमल एनाटॉमिकल वेस्ट है वो आपका रिसर्च सेंटर्स में मेनली आपको देखने को मिलेगा। सो वहां पे जो भी कॉटन, ग्लव्स, कुछ भी जो भी ह्यूमन और एनिमल वेस्ट से कंटामिनेटेड है, चाहे वो ब्लड हो, यूरिन हो, स्टूल सैंपल हो, पस हो, anything, so that can be discarded in yellow bag. Okay, and in the red bag, contaminated waste that can be recycled. Now next is white category. White categories are puncture proof, okay, leak proof, टेम्पर प्रूफ कंटेनर्स। बेसिकली ये आपके प्लास्टिक्स के होते हैं, वाइट जो कंटेनर्स होते हैं ये पंचर प्रूफ होते हैं, लीक प्रूफ होते हैं, क्योंकि अगर ये लीक प्रूफ नहीं होंगे तो जो आप इसमें शार्प्स डिस्कार्ड कर रहे हो तो क्या होगा, उससे चांस होंगे कि वो हार्म कर सके या वो कांटेक्ट में आ सके। इसलिए ये टेम्पर मतलब आपके लिए इस तरह से रखे जाते हैं कि पंचर प्रूफ भी होते हैं, वो जो हम इसमें शार्प्स डिस्कार्ड करते हैं ना उनसे वो चीजें पंचर नहीं कर सकता इस कंटेनर को, इस तरह से इनको डिजाइन किया जाता है। इसमें हम जो टाइप ऑफ वेस्ट डिस्कार्ड करते हैं वो हम इंक्लूडिंग शार्प्स एंड मेटल्स। सो निडल्स हम जो डिस्कार्ड करते हैं वो करते हैं व्हाइट में। ओके, निडल्स हो गई एंड लेंसेट्स हो गए, मेटल्स की हो गई, जो हमारे कैथेटर्स वगैरह में जो मेटल्स यूज होते हैं ना वो सब कुछ हम व्हाइट में डिस्कार्ड करते हैं। Next is blue. Blue is the cardboard boxes. Okay, with blue colored marking. Cardboard box होते हैं ये, cardboard is your गत्ते को जो बोलते हैं ना वो होता है कार्डबोर्ड, कार्डबोर्ड बॉक्स होते हैं ये, ब्लू कलर की मार्किंग होती है इन पे हम बेसिकली ग्लास वेयर्स डिस्कार्ड करते हैं, दैट इज योर स्लाइड्स। ओके, जो हम माइक्रोस्कोपिक स्लाइड्स लैब में यूज करते हैं आर द बेस्ट एग्जांपल, सो वो हम इसमें डिस्कार्ड करते हैं, राइट। सो नेक्स्ट, यस दिस इज द वेस्ट सेग्रीगेशन कि हाउ वी कलेक्ट वेस्ट एंड आफ्टर द कलेक्शन ऑफ वेस्ट व्हाट वी डू। लाइक येलो बिन, मैंने आपको बताया कि येलो बिन में आप कौन कौन से वेस्ट डिस्कार्ड करोगे, ह्यूमन एनाटॉमिकल और एनिमल एनाटॉमिकल वेस्ट। ओके, मेडिसिन। सो आपके अब इसमें आते हैं इन्फेक्शियस स्वैब्स मींस योर जो कॉटन पीस है या जो आप जिनसे पस सैंपल कलेक्ट करते हैं, जो स्वैब्स होते हैं हमारे इन्फेक्टेड विद ह्यूमन सैंपल्स। ओके, सो वो हम इसमें करते हैं, गॉज पीसीस करते हैं, बैंडेजेस करते हैं बिकॉज कंटामिनेटेड विद ब्लड, लाइनिंग्स करते हैं, जो पेशेंट लाइनिंग्स होती हैं, बॉडी पार्ट्स करते हैं हिस्टोपैथोलॉजी में, वहाँ पे बॉडी पार्ट्स हम स्टडी करते हैं, टिश्यूज वगैरह की तो रिसीव होते हैं या ओटी में आप कह सकते हो, तो बॉडी पार्ट्स को हम डिस्कार्ड करते हैं येलो बिन्स में। ओके, नाउ इंसिनरेशन, व्हाट इज द ट्रीटमेंट फॉर येलो बैग। इंसिनरेशन कैसे ट्रीट किया जाता है, रेज के थ्रू जो हमारा इसमें वेस्ट होता है, जो डिस्कार्ड हुआ है वेस्ट, वो डायरेक्टली क्या होता है, एश फॉर्म में कन्वर्ट हो जाता है। सो सॉलिड कन्वर्ट डायरेक्टली इनटू एश फॉर्म। ओके, एश फॉर्म में जब वो कन्वर्ट हो जाता है तो उसके बाद तो सॉइल में मिक्स हो जाता है, इस तरह से वो खत्म हो जाता है, बिल्कुल चीजें जीरो हो जाती है, जो येलो बिन में हम करते हैं। इसलिए जितना भी इंफेक्टेड वेस्ट है अगर हम उसे किसी और कंटेनर में डालेंगे तो उससे क्या होगा, वो इन्फेक्शन कभी कंट्रोल नहीं हो सकता। अगर हमने रेड बिन में डाल दिया, ब्लू बिन में डाल दिया, उनका ट्रीटमेंट का अलग प्रोसीजर है, तो उस केसेस में क्या होने के चांसेस रहते हैं कि जो हमारे जब इन्फेक्शन है वो फ्रॉम वन पर्सन टू अनदर ट्रांसमिट होने के चांसेस रहते हैं, और नॉट अ पर्सन, विद इन द कम्युनिटी ऑल्सो। लेट सपोज कि आपने हॉस्पिटल से वेस्ट कलेक्ट किया, ओके, वो किसी मतलब जिन्होंने ट्रीट किया उन्हें वो इन्फेक्शन वहां से गेन हो गया, क्योंकि हर एक कैटेगरी को ट्रीट करने का डिफरेंट टेक्निक है। सो अगर लेट सपोज कि आपने रेड बिन में येलो बिन वाला वेस्ट डिस्कार्ड कर दिया, सो उसको ट्रीट करने की ऑटोक्लेविंग एंड रिसाइकलिंग टेक्निक है। अब जब वो ऑटोक्लेव करेंगे तो क्या उनको इन्फेक्शन के चांसेस नहीं होंगे, उस केस में वो इन्फेक्ट होंगे, और वो इन्फेक्शन एक फ्रॉम वन पर्सन टू अदर लाइक दिस, वो बहुत वाइड एरिया में स्प्रेड होने के चांसेस है। सो इसलिए हमें सबसे इम्पोर्टेंट है कि वेस्ट मैनेजमेंट का ध्यान रखना कंपलसरी है, इंपॉर्टेंट है, आपको प्रॉपर बैग्स में वेस्ट को डिस्कार्ड करना है। नाउ नेक्स्ट, रेड बिन में इन्फेक्टेड वेस्ट डालते हैं हम, कंटामिनेटेड, जो कि रिसाइकल हो सकता है। तो कौन कौन सा हमने वेस्ट पढ़ा? प्लास्टिक्स वेस्ट, ओके, कैथेटर्स, ट्यूबिंग, IV बॉटल, ग्लव्स, ब्लड बैग्स एक्सेट्रा। नाउ व्हाट इज द ट्रीटमेंट आइडिया? ऑटोक्लेविंग एंड रिसाइकलिंग, क्योंकि हम इसमें रिसाइक्लेबल वेस्ट डाल रहे हैं तो रिसाइकलिंग तो होगी। ऑटोक्लेविंग का मीन्स क्या है कि आपका वो जितना भी वेस्ट है उसको ऑटोक्लेव किया जाता है, और ऑटोक्लेव का प्रिंसिपल है कि वो स्टेरलाइज कर देता है उन चीजों को, फ्री फ्रॉम डिसइंफेक्शन और एनी माइक्रो ऑर्गेनिज्म, ओके, वो डिसइंफेक्ट कर देता है चीजों को, एंड उसके बाद रिसाइकल होता है, फैक्ट्रीज में जाता है, मतलब दोबारा से वो पैकिंग होती है, क्लीनिंग होती है, हर चीज होती है, ठीक है। तो बेसिकली जो रेड बैग का वेस्ट है वो क्या किया जाता है? ऑटोक्लेव किया जाता है, करेक्ट। नाउ नेक्स्ट, व्हाइट बिन में हम क्या डाल रहे थे? निडल्स विद और विदाउट सिरेंजेस एंड स्कैपल्स, ब्लेड्स, इसको भी ऑटोक्लेव करते हैं। रिसाइकलिंग के लिए फाउंड्रीज को देते हैं, फैक्ट्रीज को, मतलब डिफरेंट डिफरेंट, जैसे सिरेंजेस है वो एक प्रॉपर उसको दिया कम्युनिटी को, फिर निडल्स है उनको तो हम दोबारा यूज नहीं कर सकते हैं तो उसको इंसिनरेट किया जाता है। इस तरह से डिफरेंट डिफरेंट उसको सेपरेट किया जाता है वेस्ट को, और फर्दर ट्रीटमेंट किया जाता है। बट फर्स्ट ऑफ ऑल उसको ऑटोक्लेव किया जाता है, पूरे वेस्ट को स्टरलाइज किया जाता है, करेक्ट। नेक्स्ट, ब्लू बिन में हम ब्रोकन ग्लासेस डाल रहे हैं, वाइल्स डाल रहे हैं, एम्पुला डाल रहे हैं। इन सब बेसिकली जितना भी वेस्ट है ना, सबको पहले ऑटोक्लेव किया जाता है। ऑटोक्लेव करने का पर्पज यही है कि जितना भी उसमें इन्फेक्शन है उसको खत्म किया जाए, स्टरलाइज किया जाए, डिसइंफेक्टेंट बनाया जाए उस वेस्ट को। उसके बाद उसकी फर्दर प्रोसेसिंग होती है, रिसाइकलिंग के लिए जो चीजें जा सकती है वो रिसाइकल्ड होती हैं, जो नहीं जा सकती है वो कुछ चीजें बर्ड आउट कर दी जाती है सॉइल में, ठीक है, मतलब कि उन्हें मिट्टी में वो कर दिया जाता है, दबा दिया जाता है, एंड कुछ चीजों को इंसिनरेट कर दिया जाता है कि डायरेक्टली ऐश में कन्वर्ट कर देते हैं, राख में, ताकि इन्फेक्शन ना फैले। एंड कुछ चीजों को अलग अलग फाउंड्रीज जो होती है, उनकी कम्युनिटी होती है, उनको हैंड ओवर कर दिया जाता है, और फिर वो अपने जो भी उनका प्रोटोकॉल होता है उससे फॉलो करते हैं, राइट। सो ब्लू बिन हमारा ब्रोकन ग्लासेस के लिए है, वाइल्स के लिए, एम्प्यूल्स के लिए। इसमें ऑटोक्लेव करते हैं सबसे पहले, फिर उसके बाद रिसाइकलिंग होती है। नाउ नेक्स्ट, ग्रीन बिन जो होता है वो हम मेनली घरों में यूज करते हैं फूड वेस्ट के लिए, ओके, फूड वेस्ट के लिए करते हैं, फ्रूट्स, सीड्स विद नो पॉलिथीन्स एक्सेट्रा, जो जितना भी हमारा फूड, मतलब सब्जियों के और फलों के, जब हम उनको पील करते हैं, जो उनके छिलके होते हैं, ये सब फेंकने के लिए यूज करते हैं, इन सब के लिए। अब गीला जो कूड़ा होता है ना घर का, वो इसमें हम डालते हैं, बट पॉलिथीन्स का यूज नहीं करते हैं, ओके। नाउ नेक्स्ट, यस, स्टोरेज ऑफ BMW। सो ये सब था हमारे कि कैसे कैसे वेस्ट का ट्रीटमेंट होता है, क्या प्रोसीजर होता है। हमने पहले देखा कि वेस्ट को कैसे डिस्कार्ड करना चाहिए, किस कलर कोड में कौन सा वेस्ट डिस्कार्ड होगा। नेक्स्ट हमने देखा कि उनको फिर डिफरेंट जो कलर कोड्स के बैग्स है उनको कैसे ट्रीटमेंट किया जाता है। नाउ नेक्स्ट, अब स्टोर कैसे किया जाता है, उन बायोमेडिकल वेस्ट को स्टोर कैसे करते हैं। उनको स्टोर करने के लिए जो लैब वेस्ट है, जिन बैग्स में लैब वेस्ट है, उनको पहले एक ऐसी जगह पे लाया जाता है, एक ओपन एरिया में लाया जाता है बेसिकली, और वहां पे लाके उनको टेम्पररी स्टोर किया जाता है, जब तक कि हम जो हमारे जिन्हें हमें हैंड ओवर करना है। मैंने आपको बताया था कि दो हमारे फैसिलिटीज है जिनको हम अपना जितना भी बायोमेडिकल वेस्ट है हैंड ओवर करते हैं। कौन कौन सी है? एक है कॉमन बायोमेडिकल वेस्ट ट्रीटमेंट फैसिलिटी, और एक है हेल्थ केयर ट्रीटमेंट फैसिलिटी, ओके। सो जितना भी हमारा लैब्स का वेस्ट है, हॉस्पिटल का वेस्ट है, उसे एक जगह पे लाया जाता है, एक सेक्शन होता है हर जगह पे वेस्ट मैनेजमेंट के लिए, तो हर जगह से कलेक्ट करके वेस्ट को वहां पे लाया जाता है और टेम्पररी उसे वहां स्टोर किया जाता है, जब तक कि वो ट्रांसपोर्ट नहीं हो जाता जहां पर उसे जाना है ट्रीटमेंट के लिए। ओके, सो फर्स्ट ऑफ ऑल सारा बैग्स, जितने भी बैग्स हैं लैब कंटेनिंग वेस्ट के, हर एक लैब से उन बैग्स को कलेक्ट किया जाता है और एक सेफ जगह पे लाके टेम्पररी स्टोर करते हैं जब तक कि वो उसकी ऑथराइज्ड पर्टिकुलर फैसिलिटीज को हैंड ओवर नहीं कर दिया जाता डिस्पोज होने के लिए। ओके, नेक्स्ट अब ट्रांसपोर्ट की बारी आई। अब हमें ट्रांसपोर्ट में क्या देखना है, ट्रांसपोर्ट में हम दो तरह से करते हैं। एक ऑफ साइट ट्रांसपोर्ट होता है, इसमें क्या होता है जितना भी हेल्थ केयर वेस्ट है उसे एक ऐसी साइट पे लेके जाया जाता है जो कि पब्लिक एरिया से, रेजिडेंसी एरिया से और हेल्थ केयर फैसिलिटी से दूर हो। क्योंकि अगर आप वहीं पे ही हम उसको अगर डिसइंफेक्ट करेंगे वेस्ट को तो क्या होगा, जो भी उसकी फ्यूम्स है या जो भी केमिकल्स हम यूज कर रहे हैं हम, क्योंकि जब हम उनको डिस्कार्ड करेंगे, डिसइंफेक्ट करेंगे तो सीवेज लाइन में या कहीं ना कहीं से वो कांटेक्ट में आएंगे लोगों के। इसलिए उनको ऐसी जगह पे लेके जाया जाता है कि जो हेल्थ केयर सेक्टर्स से दूर हो और पब्लिक एरिया से भी दूर हो, वहां लेके जाया जाता है सबसे पहली बात। सेकंड थिंग, बायोमेडिकल वेस्ट फ्रॉम स्टोरेज एरिया is transferred to the common regional facility for BMW final disposal। जो हमारी टेम्पररी स्टोरेज एरिया है जहां पे हमने लाके स्टोर किया था लैब से लाके जहाँ पे हमने टेम्परली स्टोर किया था वहां से उनको लेके जाया जाता है, जो भी उनकी एक कॉमन रीजनल फैसिलिटी है जहाँ पे भी उनके लिए स्पेस बनाए गए हैं, जो भी ट्रीटमेंट प्लांट्स हैं जहाँ उनका ट्रीटमेंट होना है बेसिकली वहां लेके जाते हैं उन्हें। और जहाँ जो हमारा टेम्पररी स्टोरेज एरिया है उसको क्लीन करते हैं जब तक कि वो उसकी ऑथराइज्ड कम्युनिटी को हैंड ओवर नहीं कर दिया जाता उसके फाइनल डिस्पोजल तक। ओके, सो हमें दो चीजों का ध्यान रखना है किस तरह से स्टोर करना है, कि फर्स्ट ऑफ ऑल हमें टेम्परेरी प्लेस पे स्टोर करना है उसे जब तक कि वो ट्रांसपोर्ट नहीं होता, और प्रॉपरली स्टोर करना है। हमें कभी भी इस तरह से स्टोर नहीं करना कि उससे और लोग इन्फेक्ट हो। तो हमें कुछ चीजों का ध्यान रखना होता है उस ड्यूरेशन में। आपके जो बैग्स है वो मतलब प्रॉपरली सील्ड होने चाहिए। ओके, एंड कहीं से पंचर प्रूफ होने चाहिए, मेन बात कहीं से लीकेज नहीं होनी चाहिए, कोई प्रॉपर डिस्कार्डिंग होनी चाहिए उनमें, एंड बार कोड्स वगैरह भी होने चाहिए, वो सब अभी हम पढ़ेंगे। उसके बाद ट्रांसपोर्ट तक उसे हमें प्रॉपरली मेंटेन करना है, स्टोर करना है। फिर उसके बाद ऑफ साइट ट्रांसपोर्ट होता है, पहले की जो हेल्थ केयर वेस्ट है उसको पब्लिक स्ट्रीट से दूर ले जाया जाता है ताकि जो भी आसपास के लोग हैं वो इन्फेक्टेड ना हो, और जितना भी सारा हमारा वेस्ट है वो स्टोरेज एरिया से ट्रांसफर करके किसे दे दिया जाता है जो हमारी ऑथराइज्ड कम्युनिटी है उसके फाइनल डिस्पोजल के लिए। सो इस तरह से हम स्टोर करते हैं वेस्ट को। नेक्स्ट, अब कुछ इंपॉर्टेंट रूल्स हैं बायोमेडिकल वेस्ट के लिए, बिकॉज ऐसा नहीं है कि हम अपनी मर्जी से कहीं पे भी डिस्कार्ड कर देंगे वेस्ट को, या किसी को भी हम ट्रीटमेंट के लिए दे देंगे, नहीं, उसके प्रॉपर कुछ रूल्स होते हैं। बेसिकली ये यूज होता है कि जो हमारे वैक्सीनेशन कैंप होते हैं, ब्लड डोनेशन कैंप होते हैं, सर्जिकल कैंप्स होते हैं, वहां से भी कुछ वेस्ट प्रोड्यूस होता है। जब ब्लड डोनेशन कैंप है वहां पे भी नंबर ऑफ वेस्ट यूज हो रहे हैं, जैसे कि कॉटन वहां से एज अ वेस्ट प्रोड्यूस होगा। ओके, जो ब्लड बैग्स के आगे ट्यूब्स है, ब्लड बैग ट्यूब्स है, कैथेटर्स है, वो सब चीजें भी यूज हो रही है। सो देयर आर डिफरेंट वैक्सीनेशन से आपके सिरिंजेस एंड मेडिसिन और ये सब भी जनरेट हो रहा है। ओके, सो डिफरेंट कैंप्स और हेल्थ केयर सर्विसेज से वेस्ट प्रोड्यूस होता है। तो उन केसेस में क्या होता है, यूज करते हैं क्लोरिनेटेड प्लास्टिक बैग्स का, ग्लव्स एंड ब्लड बैग्स विद इन 2 years। ओके, एंड प्री ट्रीटमेंट करते हैं हम लैब वेस्ट का, जितना भी लैब से वेस्ट जनरेट होता है उसको प्री ट्रीटमेंट किया जाता है। मैंने आपको बताया ऑटोक्लेविंग, सबसे पहले ऑटोक्लेविंग की जाती है हर वेस्ट की ताकि वो स्टेरलाइज हो जाए। उसके बाद उसे बाहर हम हैंड ओवर करते हैं, जिन्हें ट्रीटमेंट के लिए जो हम हैंड ओवर करते हैं वो उसके बाद करते हैं हम हैंड ओवर ऑटोक्लेव के बाद। सो माइक्रोबायोलॉजी कल वेस्ट जितने भी होते हैं, हमारी पेट्री डिश हो गई, पेट्री प्लेट्स जो हम यूज करते हैं, उसके बाद कंटेनर्स हो गए, डिफरेंट डिफरेंट वाइल्स हो गई, ब्लड सैंपल्स हो गए, ब्लड बैग्स हो गए। ओके, सो इन सबको पहले डिसइंफेक्ट किया जाता है, स्टेरलाइज किया जाता है ऑन साइट इन द मैनर एज प्रिस्क्राइब बाय डब्ल्यूएचओ की गाइडलाइंस के अकॉर्डिंग हम उन्हें ऑन साइट मीन्स जहाँ पे हम टेम्पररी स्टोर कर रहे हैं अपने हेल्थ केयर में, जो भी हमारी ऑर्गेनाइजेशन है वहां पे हम उसे टेम्परेरी स्टोर कर रहे हैं, तो वहां पे हम उसे पहले स्टेरलाइज करते हैं। करेक्ट। नाउ नेक्स्ट, सो हमें कुछ चीजों का भी ध्यान रखना है, ये भी हमारा एक रूल है उसके अकॉर्डिंग। फोर्थ है कि हमें जितना भी हमारा हेल्थ केयर स्टाफ है, जितने भी हेल्थ केयर वर्कर्स है, फ्रॉम लोअर डिवीजन टू अपर डिवीजन, सबको हमें ट्रेनिंग टाइम टू टाइम प्रोवाइड करनी चाहिए कि किस तरह से उन्हें वेस्ट को मैनेज करना है, किस तरह से डिस्कार्ड करना है, एंड इम्यूनाइज, मतलब उनके लिए वैक्सीनेशन कितना जरूरी है, इम्यूनाइजेशन कितना जरूरी है, ये चीजों का हमें उन्हें ट्रेनिंग उन्हें प्रोवाइड करनी चाहिए अपने हेल्थ केयर स्टाफ्स को। ओके, इस्टेब्लिश करना चाहिए बारकोड सिस्टम को। अगर हम बारकोड सिस्टम नहीं लगाएंगे तो क्या होगा कि जब वेस्ट आपने डिस्पोज कर दिया, फाइनल डिस्पोजल के लिए चला गया, वहां जाके सिर्फ आपके ऑर्गेनाइजेशन का वेस्ट नहीं है, वहां पे डिफरेंट डिफरेंट ऑर्गेनाइजेशंस का वेस्ट है। अब उस कंडीशन में जब वो सेग्रीगेट करेंगे, डिफरेंट मतलब वेस्ट को डिफरेंट डिफरेंट कैटेगरी में डिवाइड करेंगे, अगर वहां पे प्रॉपरली वेस्ट डिस्कार्ड नहीं हुआ होगा तो वो आइडेंटिफाई नहीं कर पाएंगे कि किस ऑर्गेनाइजेशन ने वेस्ट को मतलब प्रॉपरली हैंडल नहीं किया, डिस्कार्ड नहीं किया। तो इन चीजों का ध्यान रखने के लिए भी अपने ऑर्गेनाइजेशंस के बारकोड सिस्टम होने चाहिए, एंड जो मेजर एक्सीडेंट्स होते हैं उनको रिपोर्ट करना चाहिए। लाइक अगर रेड बैग्स में निडल्स वगैरह डिस्कार्ड करी हुई है तो वो एक मेजर एक्सीडेंट है, क्योंकि रेड बैग तो एक पॉलीथीन है, वो तो अगर उसमें सिरिंजेस हैं, निडल्स हैं, तो वो लीक होने के चांसेस है। सो इस तरह की कोई भी मिस्टेक जो है वो हमें रिपोर्ट करनी होती है। उसके बाद BMW 4 कैटेगरीज में क्लासिफाइड है इंस्टेड ऑफ 10। ओके, 10 कैटेगरीज होती है मेनली, बट अभी मेजरली 4 में ही डिवाइड किया है WHO ने, जिससे कि ज्यादा अगर नंबर ऑफ बैग्स होंगे तो ज्यादा नंबर ऑफ कंफ्यूजन को क्लियर मतलब कंफ्यूजन रहेगी, तो उस कंफ्यूजन को क्लियर करने के लिए इम्प्रूव द सेग्रीगेशन ऑफ वेस्ट, देयर आर 4 टाइप्स ऑफ कैटेगरी: येलो, रेड, white and blue. Okay, blue is for broken glass waste, white is for sharp metals and needles, red for contaminated infectious waste and yellow for anatomical waste. Okay, and हमारी फैसिलिटीज होती हैं CBMWTF, जो मैंने आपको बताया कि कॉमन बायोमेडिकल वेस्ट ट्रीटमेंट फैसिलिटी है, ये अवेलेबल होती है आपकी 75 किलोमीटर पर, एवरी 75 किलोमीटर्स पे ये स्टैब्लिश होती है। ओके, एंड जो CBM WTF कम्युनिटी है वो उसकी रिस्पॉन्सिबिलिटी है कि वो टाइमली वेस्ट को कलेक्ट करे। ओके, कलेक्शन ऑफ बायोमेडिकल वेस्ट फ्रॉम HCF, जो हेल्थ केयर फैसिलिटीज है वहां से उसको टाइमली कलेक्ट करे। अदरवाइज क्या होगा कि वेस्ट कलेक्ट होता जाएगा होता जाएगा होता जाएगा, और उससे जितना लंबे टाइम के लिए वेस्ट को स्टोर करोगे तो रेट ऑफ इन्फेक्शन होने के चांसेस उतने ज्यादा हो जाएंगे। इसलिए CBMWTF कम्युनिटी का काम है कि वो जल्दी टाइमली मतलब मेडिकल वेस्ट को हेल्थ केयर फैसिलिटी से कलेक्ट करे और असिस्ट करे, ट्रेनिंग दे हेल्थ केयर फैसिलिटर्स को। अगर कुछ चेंजेस है या टाइमली वो ट्रेनिंग प्रोवाइड करेगी किस तरह से उन्हें कब वेस्ट को कैसे डिस्कार्ड करना है, जो भी गाइडलाइंस है उन सबको वो ट्रेनिंग प्रोवाइड करे। सो थैंक यू सो मच।
Hello students, welcome to the study portal. So today we are going to study biomedical waste management. You are having a subject, right? So in which we are going to study unit 2, where we will discuss about the wastage, types of the waste, and we will discuss the management of the biomedical wastage. We will discuss the industrial waste. Everything will be we are going to cover in this video. So here there is categories of the waste, right? I told you we will discuss the types of waste, right? So here are the two types of the categories, which is solid waste and wet waste. In the right, so in the solid waste there will be household waste can be there, factories and hospitals waste can come under this solid waste, right? Because solid waste can be everywhere, in the hospitals also, in the industries also, and even in the houses, right? So except solid waste are only solid or semi solid, right? So these solid or semi solid can be dry or wet, fine. So the waste can be dry or it can be waste. Example, in our household, right, in the houses there will be solid waste or there will be dry waste, right? So in the solid waste there will be two types. Some will be like wet and some will be liquid types, and some will be pure solid waste, right? So there is right, so except solid waste there are only solids or semi solids, fine. So wet waste, we now coming on the wet waste. So basically any dissolved liquids based on based waste or sludge coming from the waste water plants, households, etc. come wet waste is called as wet waste, right? So examples, leftover curries, juice, rotten vegetables will come under this wet waste. All the things which are coming under the wet is called the wet waste, fine. So it is about the categories of the waste, where we learnt two types of categories, solid and the waste. Now there is dry waste. There is dry waste also instead of solid, wet, because solid is two types, right? Semi solid it can, but you cannot say proper dry. So here dry waste, in the dry waste, so there will be dry waste which isn't dissolved in any form or in the liquid form comes under the dry waste. The things which will not be dissolved in any liquid, right? So we will call it dry waste, like plastic bottles. These will come under the dry waste. Now coming on the biodegradable waste and non-biodegradable, right? So so we studies first type solid waste, then second liquid waste, right? So now here we studied dry waste. Now biodegradable waste and non-biodegradable, what are these? So biodegradable waste are the organic materials, right? It can be synthesized into carbon dioxide, water, methane or organic molecules by organisms in the soil come under biodegradable waste, right? So so the things which can be synthesized into the carbon, water, methane or organic molecules by the organisms, we will call it biodegradable waste. It will destroy, right, naturally. So that is why it is biodegradable. It will combine in the soil, and it can right, and it can also synthesized into the water. So now coming on the non-biodegradable waste, so these are the materials which cannot be synthesized into the CO2, water or CS4 come under non-biodegradable waste, right? So basically these waste in the non-biodegradable waste, what happens, they will be remain same as it was, like plastic. This is a non-biodegradable waste. You cannot burn plastic, you cannot even dig, you cannot plastics cannot mixed in the soil also, right? So you cannot remove plastic, so that is why it is a non-biodegradable waste. You can see here canes, right, and here these fruits, vegetable, table scraps, these are the biodegradable. Now coming on the types of the waste, right? So there are some three kinds of waste. First is the municipal waste, and second is the household waste, right? Then commercial waste will be there, and demolition waste. So then hazard waste will come. So now coming on the household waste, so basic this municipal waste, right? So municipal waste, they are commonly consist of items we use on an everyday basis, then dump it, right? So we are using some items every day, right? So we will dump it somewhere, like clothes, pant, wires, glasses, unwanted foods, etc. come under the municipal waste. And there in the society there will be a big dustbin where we will put all these regular basis wastes, right? So these waste come from the schools, factories, but primarily come from our homes, right? So primarily these waste come from the home, and also from the school, factories, or these waste will come. So the composition of the municipal waste differs in each municipality and keeps changing with time. So municipal waste divided further into household, because these municipal waste basically it will differ, right? There will be types of wastes. So here that is why it is written as A here. So it is first one is the household waste, right? So in the household waste, what will be there, like materials like unused food and clothes, unwanted paper, damaged batteries, etc., they will come under the household wastes. Now agriculture wastes also come under household wastes. So all the agriculture wastes, these will be coming in the household wastes. Now commercial waste and demolition waste. So what are the commercial waste? So the wastage which will come from kinds of the businesses, trading factories, schools, etc., come under the commercial waste. So the waste which is coming from the schools, it can be anything, right? And from the business, and also from the factories. From the factories there will be chemical, and also these will be coming in the commercial waste. So now demolition waste. What is demolition waste? So you can see here, demolition means these waste come from the destruction any structure made of concrete, wood, brick, etc. Like somewhere there will be construction work is going on, fine, so there will be many waste will be there, fine. So these wastes will come under the demolition waste, where the destruction works are going on, construction works are going on. So destruction works basically they will there will be some waste, wooden waste will come there, bricks will be there, right? Because basically we are destructing something, so we will get many waste from there. So these will come under the demolition waste. And although sometimes demolition waste can also be recycled, and sometimes these waste can be recycled, means we can use it again. If something we are using and if it is recyclable, so we can recycle it and we can use it, because and doing recycle, it will be eco-friendly also, like, right? So if there is possibility for recycling, then we should always prefer this, and we can recycle the waste, fine. So now there is hazardous waste. So what are these hazardous waste? So these hazardous waste refer to solid, liquid or gaseous waste from industries that have either of the properties like corrosive. Corrosive means if they are having any junk, corrosiveness will be there. Ignitability, right? Reactivity, they can react to anything. And ignitability means they can they can catch fires also. And toxicity, they can produce toxic substance which will be harmful for us, like so. These types of waste, these are the properties of the hazardous waste. So we have to always keep in mind while dealing with the hazardous waste. So treatment of these waste is necessary before the industries dump it, right? So these waste treatment is necessary. Treatment means when we are dumping it, so there are some rules how we will dump it, right? It is not like that we are using any chemical, so we are just putting this chemical in the normal dustbin. No, because it is reactive, and even it can cause some infection to the persons also, to animals also, right? So it can cause any type of infection or any kind of diseases, right? Skin diseases also. So we have to treat first the waste which are hazardous waste, right? Before dumping it, we have to treat it, and there are some rules, we have to follow that rules, and accordingly we will dump this hazardous waste. So basically these hazardous waste, they are unsafe for the human health and the environment at large. So these hazardous right, so what I told you, these hazardous waste is not safe for the people, right, human health, and also for the environment. Even they can cause environmental pollution also, right? So they and it the waste, the chemicals, they will be mixed into the air, then we will inhale that unhealthy air, then that chemical automatically will come into us, right, through the nose. So that is why we avoid these that is why there is a rules for of treatment before dumping, right? So these hazardous waste, basically they are divided into further divided into industrial and the biomedical waste. So now the treatment of these waste is necessary before dump because it is unsafe. Fine. So here you can see industrial waste. So industrial waste, there are waste produced by the industries, includes any material that isn't useful for the industrial manufacture process. So how industrial waste will come? When there is a material which is not useful for the industrial manufacture process, they are not going to use it, and these wastes, such as they can be any chemicals, they can be any pigment, they can be ashes, metals, etc., come under industrial waste. So these will come under the industrial waste, right? So now coming on the biomedical waste. So any waste coming from the medical facilities such as hospital, medical college, research centers, etc., they will come under the biomedical waste. So from the hospital, what kind of waste can come? From their cottons, bandages, or syringes, all these things can come, right? From the research center, from their bottles, pen drops, all these things can come. Right, so these are the biomedical waste which is coming from the hospital, medical college, and the research centers. Fine. Now we will talk about the disposal. So the collected containers of different wastage should be handed over to the waste disposal agencies to provide specialized services of environmental friendly disposal through insertion of or dumping, right? It means when we are the containers which are collected, there will be different types of the wastage in the particular, right? So when we are dumping it, we have to keep in our mind which kind of waste we are dumping in a particular waste disposal. Fine. If it is a wet, so there will be a different disposal. If it is dry, there will be different. If it is something like toxic, there will be different type of disposal. And if there will be anything which can do harm, like needles, etc., which can cut our hands, anything, right? So they will be having a different disposal, right? So these right, so the disposal agencies will provide different types of the containers, and then we will put our waste in that. So however, it is always helpful if the waste containers are properly labeled and carry other mandatory instructions. So it will be always helpful if we are if like our waste containers, they will be properly labeled, right? If they're if we are dumping any wet, so there should be labeled, we have to put dump wet dump here. And if it is something for the for the medical purpose, so there will be types of, right? If we have to put bandages, so there will be other type of, if there will be tissue or something bloody materials, so there will be different types of the waste. So the labeling is very mandatory for this waste disposal containers, so that people will not get confused in which they have to put their types of dump, right? So that is why these labels are important in the in the waste disposal containers. So here, laboratory wastes are potentially hazardous as they can have the following characteristic. Hazardous means unsafe and harmful for the environment, right? So why they are, as I told you in previous slide also, because they have corrosive nature, right? And they have flammable, and they are highly reactive, toxic, right? They can produce some toxic substance also, and they are radioactive and biohazardous, right? So these are their characteristics which is harmful. And here you can see this is a waste from the laboratory where the Appendroff tubes are there which are used and which are not being further used, so they have disposed. So this is the laboratory waste. Now coming on this, so laboratory waste can result from several sources, of which the common ones are, so actually these wastes can be expired and unused reagents and the chemicals. As we are doing any experiment in the laboratory, so we can see in our laboratory there are many chemicals, right? So if there is some chemicals which is not in the use, they are expired, or they are like they're expired, or they are not they are unused chemicals which are not in the use, or they are destroyed, fine, so we can remove it from our lab, so they will come in the waste, laboratory waste. And there will be some reagent bottles which will be corrosed, or which will be not like they will not be good enough for other purpose, like for further experiments. And there can be some broken glasswares and plasticware items, right? Some broken glasswares should not keep in the laboratories because they will be not in the use, and then they will just take the space, fine. So for clearing the space we remove them, right? And there will be plasticware items also, which will be which is a laboratory waste. Then there is a used syringe, needle and other sharp objects, and for that syringe, needles and sharp objects there will be different type of different waste container will be there, and then we will put those into that. So now solvent used for rinsing and cleaning laboratory ware. So here we will use some solvent, right, which will be for the rinsing and cleaning for our laboratory ware, right? So these solvents will clean it nicely, and we use oil collected from pumps and generators, right? So these will come in the waste. Then supernatant from the centrifuge tube, when we centrifuge our samples, anything, so then there will be a supernatant, right? So even they will come into the waste if this is not in the use. If we are using the pallet, then supernatant is definitely like not useful, fine, so we will throw it. And body fluids and other biological waste resulting from the microbiological testing, so there will be some body fluids, right? It can be waste, blood, urine, right? Anything, so they will come in the waste, right? And there are some other biological waste, so they will be resulting from the microbiological testing. So these are the some waste, laboratory waste. And now there are recommended collection and disposal practices, right? So collection of the laboratory waste requires a knowledge of the nature of material before it is segregated and disposed of. Means when we are when we are going to collect or dispose, right? So there are some practices when we are collecting our waste, so it requires knowledge, right? Like nature of the material before we are segregating it, when, right? So we should always know the nature of the material before segregating, otherwise we can mix it, right? So to reduce the risk of mixing, the person who is collecting, so they should always know the nature, so there will be some disposal practices will be there, right? So the particular person should know their disposal practices, and then they will segregate and they will disposed of. Fine. Now acids and bases, organic waste, solvents, needle, syringe, broken glasses, supernatant, empty bottle disposals. So these are some things we should always know about, right? So there will be some ideas. Acid and basis disposal, like how we are disposing it. So for the acids we should not like we mostly drop it in the sink and we run the tap so that it will go right. And for the base also same. And then organic waste solvents. So for the organic waste solvents we will put it in a different container. And needle, syringe and broken glasses. For the needles we cannot like if the patient is having HIV, so we cannot throw it in the dustbin directly. We should always disinfect it, right? And supernatants, so we will destroy it, we will dispose it in a different one. And empty bottles, right? So these are the things how we are going to dispose it. Fine. Now waste management, now we will discuss the waste management. So basically waste management is very important, as we know, because the waste is can infect a person's health or also can harm the environment. Fine. So that is why it is important to manage it properly. And we have to we should right, because so we should always properly manage our waste. And if we are not doing it, we are improperly stored, or so stored refuse can cause health, safety and economic problems. Fine. So the living organisms create waste, but humans create for far more waste than other species. So to prevent damaging the earth's ecosystem and maintain a high quality of the life for the planet's inhabitants, humans must manage and store their waste efficiently and safely. Fine. So for our earth's health also, to maintain earth's ecosystem and quality of the life, right? For the planet's inhabitants, we should manage our waste in proper ways, right? So now coming on the biomedical waste management. So for the biomedical waste management, it is a prodigious impact, because biomedical waste can harmfully affect health, leading to serious implications to the people who get in touch with it, right? So the people who manage their waste in the hospitals and all, so or who can be in the touch, so they will have some serious problems, right? So this biomedical waste, we should always keep in our mind while dealing with it. It should be in a proper ways, right? The treatment should be proper, right? So that is why there is a rules for biomedical waste management. So here so we should know the segregation, storage and safe disposal of the waste, right? It is very crucial to effect management of biomedical waste in a workplace, right? So in the workplaces we should keep our waste in a different boxes, in a different dustbins, right? So that it will not affect the people, and also for the environment and the people who are dealing with the waste. It is it is unhealthy for them also, right? And always wear the gloves while you are dealing with the biomedical wastage. And right, so this is very important. So students, this is about the categories of the waste, and then we studied about the management. So in detail we will study in the next unit about the which type of waste. We will discuss about the types of the bags, right? There will be red, yellow, etc. types of the bags. So we will discuss in the next unit. Thank you so much students. I hope this is clear to all of you. we will meet in next video. Thank you, students. Hello students, welcome to the study portal. So in today's video we will be going to cover our topic biomedical waste management, unit 3. In the unit 3 we are going to study the color coding, where we will study the use of the different colors of the bags, and we will be studying the waste management in the hospital and what is the need for biomedical waste. We will be discussing the problem associated with the BMW and waste treatment, right? So these topics we are going to cover in this video. So let's begin. So first of all, what here is the little definition. So waste generated during the laboratory diagnosis, treatment or immunization of human being or animals, or in such activity pertaining thereto, or in the production of biologicals. So these are the hazardous waste when we are right. So biomedical waste is, so when the waste is which is generated from the laboratories diagnosis or treatment and the immunization of the human, so there will be some waste will be coming out, fine. So we will be discuss the management of these waste, how to manage, how to dispose it, everything we are going to discuss, right? So it was an introduction. Now what is the need for BMW, like why we are manage our biomedical waste, right? BMW is biomedical waste management, right? So what is the need for it? So basically there will be some flies, or there can be insects, birds, they will attract to that waste, right? So even they will get infect, and then they will be coming on us, and we will get infect. So that is why it is important. And for the filthy and unhygienic condition, if waste will be there, then conditions will be unhygienic, injury and infections. If there is like for the injuries and infection, the wound will be open, then there will be some like if there is unhygiene, or there will be some bacteria, microbes will be grow on the waste, and they will be spread in the air, and it will affect our wound which is open, right? Reuse of the disposal, and harmful chemicals and radioactive materials, they will contaminate the wells and the tanks, right? So that is why we should manage. And animals and the insects, they may get infected, or they can become carrier of the diseases, right? So and the survival and mutation of pathogenic microbes, like epidemic increase in the communicable diseases in the community, right? So for this it is important to manage the waste. And here we will be discussing the contribution of different waste type, right? How we are what are their contribution. So here the waste generation in developing countries is zero point 0.5 to 2 kg per bed per day, right? And developed 1 to 5 kg per bed per day. So here see the hospital waste. So here you can see the non-hazardous and hazardous waste. So hazardous waste will be 15 to 20%, and the non-hazardous is the 80 to 85%. And the non-hazardous waste is biodegradable. And here you can see hazardous waste is potentially infectious, toxic, and here even it is non-biodegradable or non-biodegradable. And this is potentially toxic, means there will be chemical, and it can be radioactive. So it is coming into in the pharmaceuticals, right? And there are some potentially some waste are which is potentially infectious. So it is coming from the organic or the lab, or from the shops, going on the shops, like scissors, injections, they can infect, right? And in this there will be some waste of the human body and all, they will be coming on the biodegradable. So this is about the hospital waste, where you have to learn 80 to 85% will be non-hazardous and 15 to 20% will be hazardous waste, right? Now, items and equipment which is required for the biomedical waste management. So here it is: mask, gloves, cap, boots or closed toes, shoes, right, and overgarments, apron. And here are the waste segregation bags, bins are there, which kind of waste we should put in a particular bins, right? So we will be discussing about the different types of bins also. And now here you can see weighing machine. These are some instruments which will be you can get in the laboratories or the hospitals. Here dedicated trolley where the wastage will be put, and then someone will go to throw it. And temporary central storage rooms are there, right, for the temporary storage. And scissors and disinfection solutions will be kept like this. And there is a needle cutter. You can see this needle cutter, right? So these are some instruments. And now here deep burial, sharp pit, autoclave, shredder, these things will be getting in the laboratories. Wastewater treatment plant, right? So autoclave, actually basically it is for the when you want to disinfect, like you want to kill the microbes which is present over your material, fine. So this is for that. Now, biomedical waste categories and their segregation, collection, treatment, and we will be discussing the process and disposal options. So here category is yellow dustbin, yellow bag. Why you are using it? So in the yellow bag there will be the type of the substance for that is there will be soiled waste, right? Means items which is contaminated with blood or body fluids, etc., like dressings, bandage, gauze, linen, and plaster kits, cotton swabs, bed protection pads. So these will be on the in the soiled waste, and these bags will be containing residual or discarded blood and blood components, including blood set tubings, right? And here diapers, napkins, discarded linen, mattress, beddings. So these will coming in the these are the coming in the yellow waste. So these types of the waste will be kept in the yellow bags. So here container to be used, there will be yellow colored non-chlorinated plastic bags. So it will be non-chlorinated and the common medical waste treatment facility. It is a so it will give to the common biomedical waste treatment facility, right? So now in the yellow bag, if there will be human anatomical waste like human tissues, organs, body parts, fetus below the viability period as MTP, right? Maternal trans these MTP, which is maternal transparent pregnancy, right? So termination pregnancy, so where they will be terminated their fetus in a viability period, right? And so these types of waste will also come in the yellow bag, and there will be container to be used is the yellow colored non chlorinated plastic bags, right? So you can even see here animal anatomical waste will be there. If there will be experimental and animal, so their body parts, their organ tissues, including the waste which will be generating from the animals used in the experiment or testing in animal houses. So these wastes of the animals will also come in the yellow bag, right? Or anything which is expired or discarded medicines, except cytotoxic drugs. The cytotoxic drugs will not come here, and pharmaceutical wastes like antibiotics and all also can kept in the yellow box, yellow bag, right? and their container to be used non-chlorinated plastic bags. This is for the yellow one. Now there will be some cytotoxic drugs, including all the item containing with cytotoxic drugs, along with the glass or the plastic ampoules, vials, etc. Right? So these will be coming on this type of the waste. So and there will be for that yellow colored non-chlorinated plastic bags with the cytotoxic label, right? It is not like that ki you are putting your waste in the same where you have put the anatomical waste of the animal or the uh, animal of or the humans, right? So there so you for that you will be having a separate which is non-chlorinated, but it is labeled with the cytotoxic, right? Now cytotoxic hazard symbol is there. You can see this black and then C will be there. So handle with care. This right. Cytotoxic is for the C. So we should always handle this with the care. Now here you can see some chemical waste if you are talking about. So there will be liquid waste which will be generated due to the use of the chemicals in the production of the biological and used or discarded disinfectants, right? And there will be some silver X-ray film developing liquid and discarded formalin, infected secretions, aspirated body fluids will be there. So liquids from the laboratories and the floor washings, cleaning, housekeeping and disinfecting activities, etc. So for this container will be used is separate collection system leading to effluent treatment system, right? So it will be for that. Now labs and highly infectious BMW generated shall be treated by equipment like autoclave or microwave. So which is highly infectious, we should always treat it before disposing with the autoclave or the microwave, because they will kill the bacteria and microorganisms which is harmful. Now microbiology waste and all other clinical laboratory waste shall be pre-treated by sterilization to log 6 or disinfections to log 4, right? So as per the WHO and NACO guidelines before packing and sending to CBWTF. So here it is written as use of the non-chlorinated chemicals and no chemical pre-treatment before insertion, right? Exertion. So microbiological, lab and highly infectious waste. So we should always like keep in our mind which type of waste we are dealing with and how to treat it, right? So here autoclave conditions for the BMW handling is the it should be less than 121 degrees Celsius and 15 atmospheric pressure, right? So the it is for the 60 minutes and 135 so there will be 31 atmospheric pressure and it is for the 45 minutes. And if it is like duration is long, temperature is high 149 and 52, then duration will be less 30 minutes, right? So now biological indicator, so maintain records. So first validate with 4 biological indicators. So there will be 4 biological indicators, and then daily testing with the chemical indicator will be there, and once a week biological indicator testing should be done, right? So these are the conditions for the BMW handling. Now here you can see for the yellow category only there is the microbial biotechnology and other clinical laboratory waste like blood bags, vacuolators containing bloods, dishes and the devices used for the culture. So the things which is used in the microbiology biotechnical laboratories, so if so they have to autoclave first, autoclave safe plastic bags of the container. So there will be some plastic bags, they have to autoclave it, then they will give to the common biomedical waste treatment, right? So because these can be like infected or they can be harmful they can create some infections, right? Because they will grow some microorganisms for the vaccines also. So there will be some toxic substance, so that is why it is important to autoclave them before dispose. Now coming on the red bag category, so in the red bags what we will be what we keep. So here for the contaminated waste, like recyclable wastage, we will be generate like waste generate from the disposal items such as plastic tubings, ICD, ET, drains, rails, right, these things, right? And intravenous tubes and the sets, they will be also coming in this red bag. Catheters and cardiac cannulas, all these plastic things will be coming in the red bag. Plastic IV bottles, uro bags, and syringes, vacuuteners with their needle cuts, right? Needle should be cutted, otherwise it can harm someone. So here containers which is going to be used for the red, so there will be red colored non chlorinated plastic bags or the container, right? So now gloves, nebulization, mask, surgical disposals, needle caps, plastic test tubes, pipettes, disposal tubes, plastic trough, all these things also will be coming in the red bag and in the red colored non-chlorinated plastic bags or the containers, right? So now coming on the white category, so for the white category here you can, there will be sharps. As you discussed in the red one, there will be plastics, there will be sharps, right? Needles, all the types of the needles, cannulas, stylets, right? And syringes with fixed needles will be placed here. But in the red one, the needles will be removed if you are putting in the red one. Scalpels, blades, lancets, saws, scissors, and all these things will be coming in this white, right? And any other contaminated sharp object that may cause puncture and cuts includes used, discarded and contaminated metal sharps. They will be coming in the white category. And for that there will be container which will be puncture proof, leak proof, tamper proof containers. Why? Because they are having some needles and all, right? So if it will not be puncture proof, so they can come out from the container, so it is dangerous. So that is why the container should be harder, right, puncture proof. So now these are the some like types of the containers, right? Now alternatives. So heavy duty plastic containings, but they should be leak resistance and remain upright during use without sharp able to come out. Tight fitting puncture resistance lids and translucent, which has to be marked. Right, examples are given here: plastic bottles, empty detergent, metals, cans. These things will be coming in the heavy plastic containers. And a tin or the plastic box with a small opening at the top with a list for the close, right? So close box. And now, so you can see here locally made sharp waste containers, right? How locally they will be like made. So there will be hole, right, and flap for the closing to the hole. There will be a flap so that we can close that all, and the lid will be detachable, right? So here you can see this, and even this is also puncture proof. And these are the some needles and all they are collecting, and they are keeping it right. Now you can see this picture also, we should not do like this, and it should not be keep anywhere like this, right? So it is do's and don'ts. And no, and we should never overfill our container, like it is sharps are here, right? Because if someone will touch it, so they can be hurt, fine. So we should not full it, only 3 by 4, right? So the syringe should be either multilated or needles should be cut or stored in tamper proof, leak proof and puncture proof containers for the sharp storage. And why these we are doing? Because if someone will touch, or it can like it is harmful for them, right? They can have some injuries for that. That is why we are doing like this. Now coming on the blue bag. So here in the blue bags we will be keeping the glasswares, right? Which are the which are the not in the use, or the glasswares which is contaminated, or which is broken, right? So these uh, uh, will be keep in the blue bag. So these glasswares can be broken, petri dish, slides and cover slips can be there. Broken pipettes and other glass materials will be there, right? So metallic body implants, all these things will be coming in the blue category. So containers to be used is cardboard boxes with blue colored marking, right? There will be blue colored marking. If soiled with the infectious waste, so it will be treated by disinfectants or autoclaving, right? So this is the for the blue category. Methods of the disposal of the microbiological samples and the culture. So here you have disposal of sputum container with specimen and algae culture. So you will be seeing here sputum media. Then the container is will be autoclaved, right? And where it will be put, it will be clean with the soaked in the container of 5% alcohol overnight, right? Then the container is autoclaved, and then the phenol solution colored off in the drain, and the container is sent to the final dispose site in the red bag, so that all the problem or the disease which is in the sputum, they will be like finished, they will be destroyed. Disposal of the blood samples. So here blood samples and the containers will be there, and there will be some clotted blood samples along with the container, right? So for that here you can see the 10% sodium hypochloride we will be treated, and then the container sent to the disposal site and the disinfectants to the drain. And for the clotted one we have to autoclave it, then container sent to the common disposal site in the appropriate bag, right? So other infectious samples, they will be urine, pus, stool, sputum. So they will be treated with the 5% phenol, then they will be keep for the disposal purpose, right? They will be kept in the particular bag. Now coming on the disposal of the microbiological cultures. So here these are the cultures, 10% sodium hypochlorite overnight, and here there will be autoclave bags, right? So will and discharge into the drain, and petri dishes, autoclave if usable, disposal site for the recycling. And autoclave and the reuse of the petri dish and the culture medias can be disposed of into the drains. So why we are autoclaving? Because they will be having many bacteria, fungus, because we are growing in the fungus bacteria in our petri plates, right? So if we will dispose it in a simple way, then it can be harmful for us and for the environment also, right? So that is why we do this, that is why we have this big process of biomedical waste management. Now transportation. So always collect the waste in covered bags, right? So our bags will be covered in the bins or the bags, whatever, right? And bags should be always filled 3 by 4, because dispose cannot be come out from the bag or the bin. And we should always hand over the waste to the dedicated waste collection team, right? It should not be like the simple one, so that they will not deal with it in a serious manner. So collection team should tie properly labeled bag weight record particular in waste collection register, right? So then the collection team should keep the waste bag container in transportation trolley container, and the transportation should be done safely without spillage and leakage. Now the bag should fill like this, and it should be it should be tightly packed, right? So the collection container should then be cleaned with the soap and the water. So after collecting, it should be cleaned with the soap and the water, so that any disinfection or any um, like uh, any spoiled material will come out from this, and it will be clean, right? So then new suitable bag or container will be applied and to be checked by the by staff nurse or technicians OT laboratory of the respective areas, right? And no waste to be stored uh, like uh, um, like uh, greater than no waste should be stored 24 hours of point of the segregation. So if we can only keep it till 24 hours, after 24 hours after the segregation we should not keep it, we should always dump it. And collection team then transport waste in a closed container to storage area in a predefined path of least crowding. So where the crowd is come, all the wastes will be handled there. So waste in common storage area will be delivered to the authorized person from common biomedical treatment facility, right? So it is everyone's responsibility to keep the wastage in a particular dustbin, as we have as we know that even in our homes there will be two dustbin, one for the plastic and other things, and one is for the kitchen waste, right? So we should always keep in a different containers, so that there will be no chances of the diseases, or for the persons who are segregating the waste further, right? So it is important because these kitchen wastes are the biodegradable. If we will combine it with the non biodegradable, it is only affect our environment, right? So it is everyone's responsibility to keep the wastes in a different containers as it is given. And the biomedical waste management is very important because it is somehow it affects the human health and also the environmental health, right? So this is, I hope in this video it is clear to all of you which type of waste will be kept in which type of the container, right? And also the transportation, and also it is clear to all of you like hospital waste management how to do. Fine. So I hope this video is going to be helpful for you. So thank you so much students for watching this video. Thank you students. Hello students, welcome back to the study portal. So today we are going to study biomedical waste management unit 4, where we are going to discuss the biosafety in the operation theater, and also we will be discuss the safety of the operation theater equipments also, right? So it is all about the biosafety in operation theaters. So let's begin. So first of all, there are some key recommendations of CDC and HICPAC. So the full form of the CDC is Centers for Disease Control and Prevention, and HICPAC is Healthcare Infection Control Practices Advisory Committee, right? So so there are some key recommendation of these CDC and HICPAC, where what is coming first: ventilation and water system. performance, right? So there should be ventilation and water system should be good in the hospitals, and there should be multi disciplinary risk assessment team should be there, right? And use of dust control procedures during constructions, repair, renovation or demolition, right? So when there will be some repair or construction work is going on, so there will be so much of dust will be produced, right? So there should be some dust control procedures during these time, so that the patient will not get harm. And there should be environmental surface cleaning and disinfection strategies geared towards the antimicrobial resistant organisms. So why it is? Because there will be some microbes and organisms will be there, right? So it is important for the disinfection process, otherwise it can harm the particular patient, right? Now coming on the regulation of the operating room environment, right? So there should be ventilation because it is for the positive pressure, right? And there should be a variable air system, it is also because of the positive pressure only. And air introduced at the ceiling and exhausted near the floor. So air should be introduced at the ceilings, and there should be exhaust near the floor. So that is why if air which is inside the it can go outside, right? So for this exchange of the air it is important. And humidity should not be more than 68% in the hospital. It is always less. And temperature control should be there, so that if there will be high temperature, so it can be controlled, or if there will be low temperature, so it can be controlled, right? So these are some important things which is very important in the operating room, right? Because operating room's environment should be maintained. Now, can we sterilize operation theater? So yes, sterilization means eradicating germs completely, right? So we are destroying the germs completely, and which is not 100% possible in an operating theater, because it is not possible 100%, but we can do it, right? So the source of the bacterial contamination are from air and the environment, infected body fluids, patient articles or equipment, right? So these things will create these things are the sources for the bacterial contamination, and it will be present in the operation theater, right? So yes, we will try to eradicate the germs, but it is not like sure for 100% we can do it, right? Just because of there will be some sources will be present through which the contamination can come. Now follow the standard protocols. So the one who is working in the operation theater, they will have to follow the protocols, which is containing there should be a standard cleaning and disinfection with appropriate chemical agents, and there should be good theater practice, and discipline can provide a microbiologically safe environment, right? So the environment should be microbiologically safe, because so that the patient is not getting any infection. Just because of that, environment should be clean, right? And also the who are working in the operation theater, for them also the clean environment and micro free environment is mandatory for their health also, right? So there are some precautions which we have we use for the for reducing the rates of the infection, right? So here, so every hospital must establish an infection control committee. So it is saying that every hospital should have should have an infection control committee, and it will monitor the events in the hospital on all matters related to the control of the infection. So the committee will be responsible for the things, right, for the particular things. So they will have take care of the things, right? So they will always try to try to do their best so that there will no infections will spread or no infections will be there, right? They will control the infections. And the second point is the entry of the unnecessary personnel should be restricted into operation theaters, as everyone potentially contributes to infection, right? It means the unnecessary persons should be restricted, right? The whoever is not have any work, so they should not come in the operation theaters, right? Now, next is the a good hand washing and monitoring reduces infections. Yes, if we are washing our hands and we are monitoring nicely, then it will reduces infections, right? So, through washing with warm water, so we should always use the warm water and a good detergent. It will bring more of an overall improvement, that is solely decontamination, sterilization with other chemicals, or fumigation. Fumigation is a sprayer, right? So through which sanitization will be easy, and all the microbes will can kill through the fumigation process. And here, frequent monitoring and training of the medical and paramedical staff must carry high priority. So they should be trained how to maintain the hygiene and how to maintain their operation theater infection free. And don't merely observe mechanical and chemical methods. So we should not merely observe this mechanical and chemical methods, right? So now, hand washing is a boom to safety. So throughout washing and carbonizations, so if done every day after the surgeries, yes. So if we are washing and carbonizing, so if it is done after every surgery, so will greatly enhance the safety standards. So it will be enhancing the safety standards, and it will reduce the repeated expenditure on fumigation, right? So if there will be in the fumigation there will be some expenditure, right? So if they will wash their hands and they will carbonize, then it will be helpful for them to reduce the expenditure on the fumigation, right? So now regulate the operating room environment. How we will regulate the operating room environment here? So we will minimize the traffic during operations, right? So we will minimize the unwanted people in the operation theater, right? And we should not keep the washrooms and all near the operation theater or in the operation theater. So basically it will reduce the traffic. And here, surgical attires. So there should be a particular surgical attires which will be in the operation theater only. You are not allowed to move it out of the operation theater, right? So there are scrub suits, cap hoods, which you can see here, cap hoods, and shoe covers are there, so that if any infection is there in the shoes, so it will be away from the operation theater. And masks, gloves, and gowns will be there, right? So these are the surgical attires, right? Now, surgeon and the skills, right? So there will be some technical skills set, excellent eye-hand coordination. So their eye-hand coordination should be excellent, and there will be extensive medical knowledge for the surgery, right? And expertise in the anatomy. So it should be he should be a expertise, he or she expertise in the anatomy, because after all they are dealing with a life, right? So the non-technical skill set, situation awareness always be there, decision making, communication and teamwork should be there, leadership quality should be there. So this is the skills which is required for a particular surgeon, right? Cleaning and disinfecting. So environmental surfaces. So now coming, so environmental surfaces should be always clean, and there will be medical equipments, right, and it should be disinfected, so that because they are using that in for the human body. So these knobs, handles, equipments such as x-ray machine, instrument cards, housekeeping surfaces, so these should be like uh, uh, these should be disinfected, right? So and floor, walls, chairs and the table tops all should be disinfected. They should not carry any type of bacteria or microorganism, right? So now coming on the medical equipment, so here manufacturers recommendation for the sterilization, right? So manufacturers will recommend it for the sterilization, and there will be some recommended chemicals, which particular chemicals should use for a particular instrument, and they will be water resistance properties, and also they are requiring decontamination after servicing, right? And there will be non critical medical equipment, right? So in the intermediate level disinfection it will be shown. So we should always clean our medical equipments before using, and cleaning and disinfecting. Now housekeeping surfaces, right? So frequent hand contact, high touches surfaces like doorknobs, bell drates, light switches, or wall areas around the toilet, edges of the privacy curtain, and clean disinfection more frequently. So they should be cleaned more frequently, right, every day, because it can through these infections can come into the operation theater. And minimal hand contact, and hand contact should be minimal, and hard surface floors and window scales, right, they should be clean on a regular basis, and when soiled, and when patient is discharged from facility, walls, blinds and window curtains, right, so they should be clean when easily soiled. If there will be, like, you can see some soil on the surfaces of these, so you can clean it, right? So these are the cleaning and disinfecting, like how, why we are doing this. Now cleaning, a form of decontamination by removing organic matter, salt and visible soils. So in the cleaning, what is coming, why we clean, basically if there is any matter, any salt or any visible soil, so we will just clean it, so that there will, so we can reduce the chances of the infection, right? And if there is any physical action of scrubbing with detergent or surfactants and rinsing with water, right? So these are some physical actions, and we take some necessary steps which is prior to sterilization or disinfection, right? So what is the cleaning strategy, like how we are cleaning? So to minimize the contamination of cleaning solutions and tools, like we'll be having some chemical cleaning solutions and tools, right? So change cleaning solution frequently. So our cleaning solution should be changed frequently. It is not like that if we are using it today, so we can use it tomorrow. Not like that. We should always change it within. If you have cleaned a floor with this cleaning solution, then you should change for the next one, right? So and here we should always replace soiled clothes and mop heads with each cleaning solution change, right? And launder clothes and mop heads after use and allow to dry before reuse, or use disposal clothes and mop heads. So we should always replace our soiled clothes and mop heads, right? And here launder clothes and mop heads after use, and allow it to dry before you are going to use it, right? And we should always use manufacturers recommended concentration. Fine. Now cleaning carpeting and cloth furniture. So we should always clean the carpet and cloth furnitures, furniture's cloth right, so that because it is from many days, so it can contain so many germs, microorganisms, and these things will can have infections also. So, and there is a note which is we should always avoid use of the carpeting and cloth furnishings in areas where spills are likely to occur, right? So now coming on the disinfections. So in the disinfections, so this is the process which will eliminate many or all the pathogenic microorganism on inanimate objects with the exception of bacterial spores, right? So there will be three levels of disinfection, which is high level, intermediate level, low level, right? So for the treatment of the devices and surfaces that do not require sterility for safe use, right? Now the next is the high level disinfections. So what is here in the high level discussion will be discussed. So it is including powerful sporocidal chemicals. The name are given here: glutaraldehyde or pyritic acid and hydrogen peroxide. These are the chemicals which is including here, and it activates all vegetative bacteria, mycobacterium, TB, and viruses, fungi. So it will inactivate these bacteria, right? And it is utilized for items that come into contact with intact mucous membranes and are heat sensitive, right? So here you can see semi critical medical instruments, thermometers, vaginal speculums, sigmoid scopes. So these are very important to disinfect, right? And here intermediate level, which is including chlorine containing compounds, so like alcohols, phenols, and iodophores. Some iodophores will be there. The normal one, intermediate level disinfections. So it will inactivate the highly resistant organism which is exhibit some sporocidal activity, right? So they will utilize for non-critical medical equipments like like stethoscopes, blood pressure cuffs, dialysis machines, equipment, knobs and controls. So these alcohols, phenols, they are used for the disinfecting these normal these some medical equipments which is non-critical medical equipments, right? Now low level disinfections, so it includes quaternary ammonium compounds, some phenolic and some iodophores, and it will activates the bacteria inactivates the bacteria, fungi, and the some lipid enveloped medium size viruses, right? So it will activate their functions, and it will utilize for the items that come in contact with intact skin, such as examine tabletop, baby weight scale, blood pressure cuff, all these. And now monitor employee compliance to hand hygiene recommendations. So we should always monitor our employee so that they will take care of their hand hygiene, right? So special pathogens concern. So recent studies indicates that MRSA and VRE are the most likely transmitted either. So patient to patient contacts can be there via healthcare workers also. So these pathogens can be like transmitted through the patient to patient from the health workers and from the contaminated hands or contaminated uh, patient care equipments, right? So that is why we should always clean and disinfect these equipments, and also we should always clean our hands, right? So here the hand hygiene, we have discussed hand hygiene in a like deeply. So here we should always avoid the artificial nails. We should keep our natural nails, and we should always cut it, right? So that any germs or any bacteria will not be in the nails. There will be no dirt in the nails, right? So that is why it is important, and we should always use alcohol based hand rubs which will clean right, before and after each patient, before and after using the glove also. So we should always clean our hands by the alcohol, and we should improve our hand hygiene, right, to avoid the infections. Now what is wrong with our practices? So what can be wrong in our practices so that anyone can have infections from the hospital? So disinfectant used indiscriminately. Disinfections are not in the use like in a proper use, and unnecessary we are using it, right? These are the wrong practices. We should not use it unnecessary, and not use when needed. When you are not needed, then you should not use, right? And concentration not adequate. If your concentration is not adequate, then also it is it will not work. Like for 70% alcohol, it is for the hand sanitizer, right? So there will be 70 ml alcohol, and the remaining 30 will be water, then it will be 100, right? So so I just gave you an example for the concentration, right? Now economic considerations should be in our mind, right? Business promotions, laboratory testing, hospital conditions may not correlate. So the some wrong practices are there, we should always avoid it, right? Now economical loss. So if we will use unnecessary things, so it will be harmful for the economy, right? So money wasted on the unnecessary use, and people concentrated on the floor, inanimate objects, right? So we should always like not use our resources unnecessarily, right? So here the basic care of the operation theater. So we should always clean and dry the floor, right? And we should pay great attention to the floors, using unnecessary too many chemicals not necessary, right? So we clean the floors, so just because to remove the microbes and to remove the dirt from the floor, right? Now frequent cleaning of walls and roofs operation theater is not needed. So these walls and roofs cleaning frequently is not needed, but we should clean it one twice thrice in a week when it is required, right? And do not disturb the roof. We should not disturb the roof, so do not disturb unnecessarily when it is not necessary and you are disturbing the roof, right? So do not use ceiling fans, they cause aerosol spread. And clean only when remodeling or accumulated good amount of dust, right? So how you care for floors? So use only vacuum cleaners, because it will collect all the dirt from it in it, right? And don't broom. We should not broom it, because while brooming, the dust particles will be they can enter the air, right? They will mix in the air, and then it will cause the problem while inhalation. And as it increases the bacterial flora in the environment, so as it increases the bacterial flora in the environment, as I told you, right? So here cleaning the floor. So simple detergent can reduce flora by 80%, right? And addition of the disinfectant reduces 95%. And in the busy hospitals, counts raise in 2 hours. So in the 2 hours, we should clean it in the busy hospitals. Aldehyde free sterilization of the operation theater. So for the operation theater, the sterilization should be aldehyde free, right? So now are flowers safe in the hospital? So no. How flowers can bring some bacteria on them, right? So it will come in the hospitals and it can acquire any infection. So flowers are not good in the hospitals. So who or what is important in the prevention of infections? So uh, these for the The staffs are also important, and the chemicals are also important for the prevention of the infections, right? Staff will take care and staff will use the chemicals, and chemicals is having the disinfects and all, right? So they have the disinfection properties, right? So basically the staff should always maintain the hygiene. Staff should always take care of the instruments, take care of the OT, and they should always try not to bring the things which is not required in the hospital, right? So these things are important and they can prevent the infections, right? Remember, we are more important than many, right? So now sterilization and disinfection policies are there. So we should always create own infection control team in the hospitals, right? So the infection control team, it will decide the policies, and we should always educate our staffs methods and policies, and we should always educate them hospital safety and hygiene, and educate the staff on the few useful options. Many theoretical ideas confuse, right? So too many chemicals costly need not be affected. Fine. Importance of staff education. So this staff education is important. Specify the staff for duties and responsibilities, and we should education is a matter of continuity, right? And train under the seniors observation, train the staff with the scientific goals. So staff education is very important. So who is the responsible for the operation theater's safety? So it is for everyone, right? Each and everyone. And CDC definition of surgical site infection. So this is level of classification here. The incisional SSI, which is superficial incisional, which is skin and subcutaneous tissues, and deep incision is for the involving deeper soft tissues. Organ space SSI, it involves any part of the anatomy, organ and the spaces, right, other than the incision. Here the frequent washing of hands saves several lives universally, and prevention is primary. So we should always first step is the prevention. It will protect the patient, protect the healthcare personnel, and it will promote the quality of the healthcare. So we should always create it from the web sources, visit articles of the interest on the disinfectious diseases. So we can like search the disinfectious diseases, right? So thank you so much, students. This is all about the safety in the operation theater. Hope this is clear to all of you. So we have discussed the more about the disinfections, right? So basically when it is micro free, bacteria free, then it will be helpful, right? So that is why we have discussed more about the disinfection, because it will only prevent the infection in the operation theater. So it is the everyone's responsibility to maintain the hygiene and to follow the given rule, as I told you. And there should be a there should be there in every hospital there should be their own team, right, who are dealing with these cleaning things. Fine. So I hope this video is helpful for you. Thank you so much, students.